So I would like to come back to the topic that Sven talked about in his presentation, namely actin polymerization waves. However, I'm not so much interested here in cell division or fission, but rather in cell migration and the role that these waves could play for cell migration. However, before really entering into the talk, I would like to acknowledge the people who worked together here on this work. First of all, Franziska Lautenschläger from Saarland University, who initiated the project and then also supervised all the experiments that were performed in her lab by Luisa Stankiewicz, and then a number of other people were involved in her group and in other groups at Saarland University. I would also like to acknowledge Nicola Ecker, who did essentially all the theory and lots of the analysis of the data. And we had some contribution from people in Paris from various labs, which I don't want to detail now. So, okay, cell migration. I guess you all have heard about this, but I would like to draw your attention to the fact that there are different kinds of cell migration. So, for example, here on the left-hand side, you see a fish keratocyte sitting on a substrate, and this cell is moving quite directionally, very straight, without very much deviations to the left and to the right. On the other hand, if you look, for example, at the T cell here on the right-hand side, T cells are cells of our immune system, and you will see that this cell is also moving, but has a very different pattern. In particular, this motion is much more erratic. And the question that I would like to address is, what is causing this erratic motion? What is it driven by? And one possible explanation might, of course, be that this is just due to molecular noise that might be amplified on the cellular level. But in fact, there might be arguments to think that molecular noise might not be the only or even not the main driver of this erratic motion. And the argument comes from the following, that if you look at the T cells, what they have to do, so the T cell in this picture here, which will turn into a movie in a second, is the gray thing that you see here on the left. And then you see so-called antigen presenting cells. So these would be the sick cells in your body. And as long as they are green, they are happy and alive. But as the T cell in this comes about, makes contact with the sick cell and kills it. And then it will move on to the next cell, where it will do the same thing, and will move on and on and on. And for these cells, might be quite natural that in order to efficiently really patrol our body for sick cells, they cannot just move straight ahead in one direction. They can also not really rely on any cues, like chemotactic cues, as we've seen in previous examples. They really perform at best a random walk and then thereby try to patrol the whole environment. Importantly here is that if this random movement, this erratic movement, would be just due to stochastic effects of molecular noise, then these cells wouldn't have really a way to very much control this random or erratic movement. So the question is, maybe there's some other mechanism that is generating this movement that the cell can adapt so that in different environments it can perform different kinds of random walks. Just to give you a visual impression that these cells are really doing random walks, here we can see several trajectories. And if you just then look at the mean squared displacement of these cells, then you will see that initially there's a ballistic phase, and which then turns over into a diffusive phase, really showing that on large time scales, this motion is really diffusive. Now, I don't want to discuss the topic of random or erratic motion in the context of T cells, but rather go to a different kind of immune cells, so-called immature dendritic cells. And the reason is that these cells do not rely on adhesion in order to migrate, and therefore the mechanism underlying migration is somewhat easier to understand. In order for these cells to move, as they don't adhere to the substrate, they have to be sandwiched between two plates, and there they can move forward. And in order to do that, what Luisa and Franziska generated was a roof made of PDMS, And then you had little spacers also made from PDMS of 3 micrometers height, so that you had a uniform roof with a uniform spacing of 3 micrometers in which the cells move. And then again, here, a little bit fainter, the trajectories, but if you follow these cells, I guess you can appreciate that they also move quite randomly. However, this motion is a little bit different from the T cells that we have seen before, and that is these cells, they really have 2 different kinds or distinct types of migration. On one hand, they can move in quite persistently. So you see this cell here is moving more or less on circles. If you look at the upper cell, sometimes the radius changes a little bit and you have some fluctuations around, but on average it's moving quite persistently in a circular way. However, there's a different kind of migration, and that is where they move diffusively. And this is an example that I show you here. If you now look at long trajectories over several hours of these cells, then you will see that they switch between this diffusive kind of motion and this more persistent motion. I don't show you this on a long-term trajectory here. Here I only show you really the 2 extreme cases. And this might already remind you of bacterial chemotaxis, if you're familiar with that, where you also have phases of what are called runs, in that case, where the bacteria are moving more or less straight, and tumbles, where the bacteria are changing their direction. So here is a longer trajectory where there are these 2 phases visible. So you see in blue the persistent motion, and in red the diffusive migration. How did we distinguish between them? So we developed an objective criterion, and to that what we did is we sampled the trajectory every 3 minutes and then looked at the changes in direction of the cell steps as they go on. And if there were 2 steps following each other where the direction changed by more than 90 degrees, in that case, then we said this is the diffusive migration, whereas when it's more smoother, in that case, we call it the persistent motion. Now, we can characterize these 2 phases a little bit more in detail. So, for example, we can look at the cell speed here on the right. We see that the diffusive migration is somewhat, the speed in this regime is somewhat smaller than in the other case, although you see that in both cases, the distribution of speeds is quite prominent. What is maybe more interesting is to look at the turning angle, namely the angle from one step to the next step. And here, in the case of the diffusive migration, you see it's really more or less uniformly distributed, whereas in the case of the persistent motion, you really see a nice peak around 0, justifying this name of persistent motion. However, as you have seen already, if you look at a single cell, then it often walks in circles, whereas the data that I showed you before was obtained after averaging over very many cells. And so what we did is we fitted circles to the trajectories over as large pieces of the trajectory as we could, and then just plotted a histogram of the circles that we could get. If you do this for the persistent migration, which are depicted here on the right-hand side in the histogram in blue, then you see that there's a typical radius of about 50 micrometers, which is about 2, 3, 4 times the cell diameter. Whereas if you look at the diffusive migration, then these circles or these radii of curvature, you can still define them, but they are more evenly distributed and they are much smaller. Well, much smaller, they are smaller. And then there are these big black parts of the histogram. They correspond to what we call mixed trajectories. That is, there we could fit a circle over such a part of a trajectory that contained a part of diffusive migration and a part of persistent migration. Now, next question is, we want to understand a little bit better how does this erratic motion come up. I didn't show you data that on the end you also get diffusive behavior if you look at the mean square displacement of these cells. So what is the mechanism underlying this diffusive, this erratic migration? Well, there we have to talk a little bit about the molecular origin of the migration, and this can be found in the cytoskeleton. So we have heard already the name actin. Actin is a polymer that can assemble into linear filaments and generates these red fibers that you can see in these fluorescent micrographs in the center. There's another part of filamentous proteins that you can see in the cytoskeleton, the so-called microtubules, but they are not of importance for what is going to follow. What is important here is that F-actin can be found predominantly beneath the plasma membrane, which is separating the cell from its environment. And this is really the thing that is driving cell motility. At least in many cells, here I show you again an example of a T cell and a human neutrophil. There you see that the actin is polymerizing mostly there where you see these bright spots or these bright regions. And these bright regions, in these cases, they correspond nicely to the membrane, to the boundary of the cell. And you see that this actin polymerization is really driving the cell wall or the cell membrane forward and is thereby underlying here the cell migration. I should say, though, that in contrast to the cells that we are looking at, the dendritic cells, these two cell types that I'm showing you here, they really rely on attachment to the substrate in order to move forward. Now, what I would like to do in the following is to understand a little bit how the dynamics of the polymerizing actin can be described. And here I'm going to look at a continuum description. So I'm not interested at the mechanics or the mechanisms on a molecular level. I'm rather thinking about the continuum of actin and how is it evolving. And I'm using a purely phenomenological approach, not really looking at any of the molecular mechanisms. So we have three fields. One is the filament density and one is the filament orientation. So these filaments are elongated. So if I average over a small region of the cytoskeleton, the actin cytoskeleton in this case, then it can happen that these filaments point more or less in one direction, in which I get a large filament orientation, large polarization of the actin cytoskeleton. And if they are moving in very many different, if they're pointing in many different directions, then I get a smaller value of this P. So if I look first of all at these two fields, we can look at the time derivative of these fields, and then I just write down all the combinations in linear order and to lowest order in the gradients that are compatible with the symmetries of the system. So we have, for example, that the filament density can be degraded by the depolymerization or the disassembly of the filaments, which is here parameterized by the disassembly rate Kd. And so the second term on the right-hand side of the first equation gives you this effect. Then we have also an effect of polymerization. So the filaments are assembled at the tips, at their tips. And this is captured by this divergence of P that you see here. And this is proportional to a polymerization velocity. And then, since we have a disassembly rate, we also need to generate somehow the filaments. And this is done by particular proteins that nucleate new filaments, generate new filaments. And so these nucleator proteins, as we call them, nucleators, are described by the distribution Na, and it's proportional to the rate alpha, which gives you the nucleation rate. And so this would be the phenomenological way of writing down, the simplest way, the dynamics for the actin density. Similarly for the polarization field P, which has very similar terms. However, there's no nucleation term because the nucleators, they generate actin filaments with random polarization, with random orientation, so there's no contribution there. If you don't like this approach of writing the equations down, you can also go to a more microscopic description. And then if you coarse grain that one, you arrive at the same equations. Now, obviously, these equations would be quite boring if there was not something interesting going on with the active nucleators. And here I show you what is happening. So these nucleators are proteins that are typically inactive when they are not bound to a substrate. However, once they bind to the plasma membrane, they get activated, and then they can generate new actin filaments, as shown here in the middle. And then there's a negative feedback, how these actin filaments that now exist, they accumulate other factors that would then lead to an inactivation of these nucleators. The important point here to put in is that these nucleators are activated in a collective manner, meaning where you have already active nucleators sitting, the chance of activating new nucleators is higher. And if you write this down into equations, and this is how it looks like. So for the active nucleator density Na, we have a diffusive term. Then we have an activation term, which is a base rate, omega A. And then we have this cooperative effect that I was mentioning here with omega 1 Na squared. And Ni is the density of inactive nucleators. And so these are then turned into active nucleators through these processes. And then there's also an process that inactivates nucleators, and this is proportional to the filament density C, which reflects the negative feedback from the actin filaments on the nucleators. For the inactive nucleators, we have a very similar equation. It's just that the diffusion now occurs with a different diffusion constant, and the rates are inverted, meaning that we have really a conservation of the total number of nucleators. And for very general reasons, under these conditions, it's very likely to obtain waves. And let me just go directly to the 2D system where we just show the nucleator density here in 2D, and you see spontaneously these waves appear, they disappear. If they collide, then they annihilate each other. They can split up. Phenomena that you could also see already in Sven's presentation. Sorry for this. And at the same time, you see already that the dynamic looks really chaotic. So this already hints towards the fact that by having a purely deterministic system, sorry for the telephones, that having a purely deterministic system, you can have these already erratic motion. Now, if we want to describe cell migration, then we have to somehow confine them to a cellular domain. And this we do here by using a phase field approach, similar to what Sven has presented. So the phase field is 0 outside the cell, 1 inside the cell. The dynamics is given here, and I'm not going to give it in any detail. Maybe I just mentioned that there's one term, namely the last term of the dynamic equation for the phase field, where we see the interaction of the actin filaments through their polarization with the boundary of the phase field. The boundary is there where you have a large gradient in the phase field, where it switches from 0 to 1. And there we see that our filaments effectively push against the membrane if they're pointing towards the membrane, and they pull on the membrane if they're pointing away from the membrane. And just to give you one example, you can see here that the system develops waves, spiral waves for times that then break up. And in this way, our cells, they can move forward. And if you look at very long trajectories, then you will also see that they really perform a random motion. So here I show you just one trajectory where you see the cell form at several times, and you see again, similar to what you've seen in the experimental movies, you see these more diffusive parts, and you also see this persistent migration parts. And we can apply the very same kind of analysis that we did for the experimental movies, and see here, for example, the turning angle. Again, for the persistent migration, they are clustered around 0, whereas for the diffusive migration, they are really evenly distributed. You can't see them very well here. They're unfortunately very small. I should also mention that you have really an even, this is what these histograms show, that you have an even distribution, so you can have either motion to the left-hand side or to the right-hand side. There's no overall chirality of the system. Locally for a cell, yes, but if you look at this system overall, there's no chirality in the system. You can also look at the speed, and again, here we find that the persistent migration is faster than the diffusive migration. I'm on purpose not showing you the data in comparison with the experimental data, because the quantitative agreement is very poor, and I will comment on this in a minute. And here, another thing that I would like to show you is the radius of curvature of the trajectories. And again, here we find that the radius of curvature in the persistent migration phase is about 50 micrometers. For the diffusive migration, we could determine only very, very few radii, and they're very small. So this is why you see almost no red in here. And then there are these mixed migration patterns that we can safely neglect here. Okay, what I've not shown you, and this is then the last thing, that really these dendritic cells also present actin waves. So you see some here, it's a bit hard to see, and there are not many of them. However, if you interfere with the molecular motors that are in the system, and this is a protein that I haven't talked about at all so far, so there are proteins in the system that can connect cytoskeleton filaments and perform work by hydrolyzing ATP and thereby generate really active stresses, mechanical stresses in the system. And these stresses, they are really essential for the migration of dendritic cells. So I didn't include this in my description at all. So this is why I'm not expecting that the description that I have really quantitatively recovers the dynamics of dendritic cells. What it shows, though, is that the spontaneous waves that we see, the actin polymerization waves, they can provide the cues in which direction the cell should move. And just to show you that if we inhibit these molecular motors, that in that case we get really many more waves. I give you this picture here, and here you see that these waves are much more chaotic, much more prolific as in the other case. And so this is something we are currently working on, and how to couple the dynamics in presence of molecular motors with the dynamics that we have looked at so far, and to see how this then couples together and whether this can give us quantitative agreement between our simulations and what was found in the experiments. And with this, I want to finish and just conclude quickly that spontaneous polymerization waves can orchestrate the cytoskeleton during cell motility. This is what our calculations suggest. And I would like to emphasize that here we have a deterministic mechanism that can generate erratic motility. Since this mechanism is deterministic, by changing some parameters, you can obviously then also affect some parameters of the random walk that the cells perform. And in this way, the cells could adapt to various environments. And with this, I would like to thank you. And if you have questions, I'm happy to answer them.